Okay, so for this last part of 3.0, we're going to talk about karyograms. So you guys have already been seeing a lot of these throughout this unit. So a karyogram is just a photo that shows the chromosomes in an organism's cells organized by decreasing lengths of the homologous pairs. So this picture is a karyogram, and again, you see them ordered by length. So longest ones go first, and it goes down to the shortest. So it's just a photo of the chromosomes. So that's a karyogram. A karyotype is different from a karyogram in that it is the number and type of those chromosomes. So the karyotype for this one, you would count how many there are, so how many pairs there are. So there would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So you would say that there were 18 pairs of homologous chromosomes. That would be its karyotype. So karyogram is the picture. Karyotype has to do with the number and type of chromosomes. Okay. So again, remember from last time that the sex of an organism can be determined by looking at the type of chromosomes. In humans, this is on chromosome 23. So it could be different numbers of chromosomes for different species. But in humans, we have 23 pairs of chromosomes. Remember that in humans, XX means you're female, XY means you're male. And in other organisms, you can always identify the sex chromosomes if there's a difference in size. If some of them just don't match to each other, that's probably looking at the sex chromosomes for that organism. So some diseases are caused by chromosomal abnormalities. The most famous one or well-known one is probably Down syndrome. And so that is an inherited genetic disease that results from having an extra copy of chromosome 21. So those karyograms, the pictures that we take of the chromosomes, they can be used to diagnose Down syndrome in humans. So how you get that extra copy of 21 is a result of a non-disjunction during meiosis. And people with Down syndrome have pretty distinctive features. They have these sort of flattened facial features, the upward slanting of eyes. Some of them have these slowed cognitive abilities, and many of them have poor muscle tone. And this is caused by having an extra copy of chromosome number 21. And that occurs because of non-disjunction. So again, the karyograms can be used to diagnose Down syndrome. And karyograms can be used to diagnose any chromosomal abnormalities. This is just one of them. And what you would be looking for is that you have this extra chromosome 21. So again, remember, most chromosomes exist in these homologous pairs. So we're diploid, so we have homologous pairs of chromosomes. So each of them is going to have 2, right? So chromosome 1 has 2 of these. Chromosome 6 has 2 versions, these homologous chromosomes. Chromosome 16 has 2 versions. But for 21, instead of 2, you have this extra 1. And that is what causes Down syndrome. Again, you can look at this 23rd pair here of this karyogram, and because this person has 2 X chromosomes, you can already tell that they are a female. And because of this extra chromosome here, you can tell that they have Down syndrome. And just based from the number of chromosomes that you see on this karyogram, you know that humans have 23 pairs of homologous chromosomes, and there are in fact 23, so this is probably a human female with Down syndrome. Okay. So one of the most famous people, I guess, in the chromosome world is John Cairns. And he was the first one to really take a picture of a chromosome, and he did this in bacteria using a technique called autoradiography. Autoradiography, maybe. So what he did is he grew bacteria in titrated thymidine. So all that means is this thymidine has radioactive hydrogens on it. And he threw this chemical into the E. coli while it was dividing. This radioactive thymine, this is the T of your A's, G's, T's, and C's, was incorporated into that bacterial DNA. And if you know anything about radiation, they emit particles. I think it's decaying neutrons. So radioactive substances are going to release those radioactive particles. And so what he did is he grows the bacteria with that radio-labeled hydrogen, with that radio that ends up in your thymines, and he digests the cell and released the DNA. And he laid on top of that this sort of photographic film. And when the radioisotopes were being released from the DNA, they were hitting that film where there was DNA. And from that, he was able, after 2 months, to observe what a bacterial chromosome looks like. And again, look at this. It's a single circular chromosome. And it was one of the first times that they took pictures of chromosomes. So that ends our unit on chromosomes. So it's going to end that here.